First off, thank you for answering these questions for us. Reata's Lariat trial was a phase 2 study that tested bardoxolone methyl on patients with pulmonary arterial hypertension. How did Reata come to realize that BARD could also possibly treat lupus and scleroderma patients with associated PAH?
So there's a lot of data in the literature that suggests that the target of our compound, NRF2, which the drug induces, and NF-kappa B, which it suppresses, would be important at reducing inflammation in the setting of autoimmune disease. So NRF2 knockout studies demonstrate that animals develop autoimmune syndrome, showing that NRF2 is important at suppressing inflammation. Furthermore, some of our academic collaborators have demonstrated that a close bardoxolone analog has activity in 2 different models of scleroderma. It suppresses remodeling and collagen formation, and it also has activity in patient fibroblasts from those patients who have scleroderma. There's other data in the literature showing that our drug has activity in a model of lupus, so at reducing autoantibody load and protecting various organs from the autoimmunity. And so those were the mechanistic data. And then when we conducted our phase 2 Lariat study, some of the patients within that study had connective tissue disease-associated pulmonary arterial hypertension. And so those patients showed the largest signal of efficacy in the study. And so for all those reasons, we decided to launch an initial phase 3 study in patients with connective tissue disease-associated PAH.
Okay. The trial investigates the effects of BARD as an add-on medication to other pulmonary hypertension drugs. Is there any possibility the drug can be used as a single agent, or it will be exclusively used as a combination therapy?
We anticipate that the drug would be complementary to the available therapies. Our drug targets the inflammatory, remodeling, and mitochondrial aspects of the disease, whereas all the approved therapies directly target the vasoconstrictive aspects of the disease, and therefore our drug is addressing some of the unmet need in the patients and targeting pathways that are not directly affected by the available therapies. So therefore we think the drug will likely be used in combination with the available therapies.
What valuable information from the phase 2 trial of the trial in PAH patients will now be transferred to CTD PAH patients?
So first of all, we hope that the meaningful efficacy that we noted in our phase 2 study, and so large placebo-corrected increases in 6-minute walk distance for patients who are already receiving background therapy, that could translate. Furthermore, the novel clinical profile, so the drug has activity in those patients on top of other therapies, and it's metabolically active and doesn't have some of the side effects of the other drugs. Furthermore, we hope that just the safety profile translates. We noted no major safety signals and hope that continues in phase 3.
Okay. Why do you think it has been particularly challenging to find a drug that can help relieve symptoms and slow disease progression of CTD PAH patients?
I think most of the focus in PAH has been on the vasoconstrictive pathways of the disease, and so there's 3 major classes of drugs, and they all target the vasoconstrictive pathways. There's a recent large meta-analysis published by the group at University of Pennsylvania that compared the baseline status of patients with idiopathic disease versus those with connective tissue disease-associated PAH. And what they showed is that at baseline, the patients with idiopathic disease have much worse hemodynamics than those with connective tissue disease patients, which likely explains why the idiopathic patients respond much better to available therapies and why the patients with connective tissue disease do not. And it suggests that the connective tissue disease patients have other disease processes. They're promoting their progression. And so that's likely why there hasn't been much progress made in the connective tissue disease patients.
Because CTD PAH patients have autoimmune diseases that cause their PAH, their inflammatory disease processes often involve more remodeling than other subtypes. Since BARD addresses inflammation and not only vasoconstriction, do you believe it may provide additional benefits to this particular patient population beyond current PAH therapies?
Yes, and so through the drug's anti-inflammatory effects, there's likely 2 primary effects that could be different than the available therapies. Number 1, the drug acutely improves mitochondrial function. Mitochondria are the power plants of the cell. When patients have PAH, their bodies sense the relative hypoxia, and the mitochondria tune down their energy production, which likely explains why patients on available therapies still have a significant physical impairment. And so we think acutely the drug could improve their mitochondrial function, improve energy and physical functioning, in the longer term suppress remodeling.
As you just said, Reata recently announced plans for a phase 3 study to further assess the therapeutic effects of BARD in patients with CTD-PAH still this year. Could you comment on these plans?
Yes, so we're planning to initiate our study in the second half of this year. We plan to start screening and enrolling patients. Right now, our team is working on identifying all the clinical studies, clinical sites. We expect about 100 sites will participate in the study in approximately 15 countries.
Okay, do you know more or less how many patients are expected to be enrolled?
And so we anticipate between 130 and 200 patients.
Okay. Which of the different dose regimens so far tested will be employed?
And so we'll be using a titration scheme, and so all patients will start at 5 milligrams. Then after 4 weeks of treatment, they'll be dose escalated to 10 milligrams.
Okay. And which CTDs will be considered in this trial?
So all of them, and so we expect most patients will have scleroderma, but patients with lupus or mixed connective tissue disease will also be allowed to enroll in the study.
That's it.
All right, thank you.
Thank you very much.